quick. We've got 6 days left to cover a lot of material. There's been a tendency for people wanting to sit and chit chat. If you want to do that, leave the room. I've had some people complain they can't hear what's going on across the bar. So, from now on, you've got to talk with someone, do it elsewhere, because we've got a lot of material to cover, and I'm not going to have a lot of time to repeat very much about it. So, we got to move along. I will be cutting parts of these chapters out. There's not everything in these that we will cover or need to. So you need to pay attention to which ones of those do we need to be looking at. And button of virus, I've not got the exam created yet. You'll get them Wednesday. I don't think they'll be bad, so from the looks of what I've seen. Now this particular part, we are going to cover meiosis. Meiosis is the opposite of mitosis. We're not going to spend a lot of time on this one chapter. It's one little section. That's all we ever cover with it, because a great deal of it's repeat from mitosis. So we will go through this, go through it pretty quickly. And 2 things that I want to spend plenty of time on is heredity, transcription and translation, which is how proteins are made and how DNA is replicated. So those will be the main focus of what we're going to be doing. One of the things Amy just asked, which is a good question, when is the exam? And I'm going to show you. This exam is not comprehensive. That will be given on the day that is assigned to us, and that is done through the main campus here. If I can find out. I hate to use these websites. Can't find anything. Our page here, if you go to where it says academics, academic affairs, it's got the academic calendar. Down here it's got the exam schedule. This is what we will go by.
So this exam will be the final exam?
The next exam will be your last exam. It will be done on the day of finals. It just will not be a comprehensive time.
Will we have an hour to complete it, or?
There'll be more than that. They give 2 hours usually.
Okay, but is it going to be the same length as the other ones?
Maybe not. Maybe a little bit more, because we do have a little bit more time.
Okay.
More points, the better for you guys, because they count that. Now here is the way that for any of these, if you go to this, go to academic affairs, that's Claremont, you'll see where it says exam schedule. Take your class. They will tell you if it's a Monday class or a Tuesday, the first day of the week that it starts and the time. Now this class starts at 11:20 on Mondays. That's our first day. So if we go down here and look, start time is gonna be about right there, I think. That's the closest we've got, is 11:15. We kind of changed it here because of the fact that I had to teach this class and another one and had to scrunch them together a little bit. So I had to get time to get from upstairs to down here instead of 5 minutes.
What if with our lab, since it starts at 9:50, what would that be?
You won't have one in 11. So Monday, when we start, we should be, if we were on UC schedule, it'd be 11:15 instead of 11:20. So, here we go at, Friday, December 14th, 1:30. So you've got the week, basically. It's at the end of the week on that Friday. Any other questions? It's at 1:30.
So our final exam will be in this room still? In the same room?
Same room.
Okay.
Nothing really changes there. So that is your time. Now, we're gonna jump right in. And you can find your other exams by doing the same thing. Go to academic affairs, exam schedule for 2018. What day does your class meet on? The first time of the week, Monday or Tuesday, and then the time. My host is here, and as I say, we are going to go through this relatively quickly. We'll get everything done today on this. Meiosis is the way we make gametes. That's the big thing to remember. Mitosis gives us somatic cells. Somatic cells have a full chromosome number, full chromosome set. Mitosis produces cells that are haploid. They have one chromosome set, not two. These are things that we will get into when we get into the next unit on heredity, but you can look through those things. Heredity is just the transmission of trait. Now, heredity is coming by genes that are passed down. Remember, genes, they code for proteins, but they are what will determine heredity. And as we said, they will produce the gametes, either the egg or the sperm.
Can you put the screen down?
Well, why did you ask me that? One thing, sorry about that.
Oh.
I guess it will help quite a bit.
Just a little.
I told you about the flu on the line and a half. Hey, hey. Remember, every gene will have a particular place on a chromosome in which it will reside. Another thing to remember, the gene codes for a trait. The value of that trait is called an allele. For hair color, for example, the trait, the gene would be for hair color. The allele would be the value. Is it brown, is it black, red, blonde, whatever it is. That is the allele. The allele describes what version of the gene that you have. These are talking about asexual productions and clones. Remember about reproduction, sexual reproduction. The big benefit of that is you get an exchange. You get new genes incorporated into an organism. With asexual reproduction, it's just one individual, like a plant, that just divides itself and basically clones itself. There's no new genes coming in. The problem with that, and with that in animals, it gives the opportunity for bad genes, bad traits to be passed on and amplified. Whereas if we get new genes coming in, there's much less chance of that. And notice here, we've talked about somatic cells. Just remember, in humans, we have 23 pairs of chromosomes. Chromosomes are the same. They carry the same genes. The values of those genes are different. You get 23 chromosomes from your mother. You get 23 from your father. That gives you two sets or a diploid number of chromosomes. The two chromosomes in each pair are called homologous, which means they are the same, a homologue of each other. They will both look the same. They will both have the genes, same genes. The values of those genes may be different, the traits, the alleles, but the genes will be in the same place. This shows genes. Now, if you notice here what it was talking about, this gene right here, here's the DNA. This is very confusing here, the way they put these up. First off, this is showing you, when you see this, they use it whenever things are going through metaphase, whenever they have been copied. Actually, this is only one chromosome. It has been copied for growth, so you see two of them together. This would be from the mother, that one would be from the father. Both of those chromosomes are being duplicated, and we're going to make two new cells, so each of these would then go to different cells. So when you see this, you're actually seeing one. Remember how we said on the last test, when they're together, they're called sister chromatids, and then when they're separated, they're chromosomes.
So actually, that part on the left and that part on the right is two, or just,
Do what?
so each one of those, I mean, where
It's
it,
a chromosome, each one.
oh, okay, that is.
They're
confusing.
copied.
Okay.
It started out where you had one of these and one of those, then they copied each other because they're going to go into different cells.
All right.
This is the karyotype. It shows you what the different chromosome lengths, shapes are. This is the sex chromosome, 23rd, the big X, and then the Y. And this is confusing. It's not a good way of doing this because this is showing, like in the first phase, in the middle phase, like in mitosis, whenever they're copied and they're starting to separate, but they're not really individual chromosomes yet. We talked about the sex chromosomes. Sex chromosomes can be either X or Y. Now, a mother, a female, can only donate X's because that's all she has, right? A father can donate either an X or a Y. So it will be the father that will be the one that determines the sex of the child. The sex chromosome, remember, chromosome number 23. The other are called autosomes, which means they are basically the same. Whenever cells divide, as we will see, form gametes, one X will go into one egg, this X will go into another egg right there. So the only thing a female can give is X's. The male, on the other hand, can give an X or a Y. We know, I've talked about diploid cells. I'm not going to spend a lot of time on that. Humans have 23 pairs for a total of 46 chromosomes. That's how many they have. But remember, the chromosome number for humans, 2N. N represents one set, so that is 2N. In a cell in which DNA has been copied, each chromosome is replicated. It's what we just talked about in the interphase. Each replicated. replicated chromosome consists of two identical sister chromatids. That's what we've just talked about. If we only have one half of the chromosome number, or in one set, that cell is said to be haploid. Fertilization is when the sperm, which is haploid, unites with the egg, which is haploid. Then they make the first cell that is diploid, that has a full set of chromosomes. That first cell is called a zygote. Remember that one. After that cell is formed, the zygote, the cell will start to replicate, and it will do that by mitosis. And then it will make an embryo, eventually a fetus. At sexual maturity, the ovaries and the testes start to produce haploid gametes. Gametes are the only types of human cells produced by meiosis. Remember that. Meiosis will give us one set of chromosomes, mitosis two. This, as you can see, we've got the two gametes right here. They come together, fertilize. We've got the zygote, goes by mitosis all the way up until where it gets to be the adult stage, where it will go through mitosis for the adult then to produce the haploid cells, the sperm or the gamete. Nothing new there in animals. Keep in mind in plants it is different, or can be. Plants have a way of doing asexual reproduction in a little thing called alternation of generations, and you'll get into that whenever you get into 1082. And notice here what it's saying. You're going to hear the term spore a lot. Plants can produce spores. Bacteria can produce spore. Fungi can produce spores. So it's a common type of little structure. I'm not going to go into sporophytes and haploid things. We'll cover it just a little bit. I do want you to know what it is, but you will get into the main part when you get into 1082. So for here, alternation of generations. Notice, this is a stage. One stage can be diploid, and the other stage can be haploid. What I want you to remember, the diploid structure is called a sporophyte from a plant. It makes spores, and it does so by meiosis.
So are spores gametes?
Yes, well, no, they're not. They do the same, kind of the same thing almost.
So are gametes for animals only? That term?
Alternation of generation, yeah, pretty much, yeah. It can be fungus too, but non-animal. So sporophytes make spores. Those spores are haploid. Each spore grows then by mitosis into a haploid organism. So we don't have a diploid organism here. We don't have two sets of chromosomes. We only have one set, but it goes from a spore all the way up into this structure, a new organism called a gametophyte. Now, what do you think a gametophyte is going to produce? Gametes. A gametophyte makes haploid gametes by mitosis. It just simply starts to copy that cell again. if these gametes are fertilized, we grow a sporophyte. Now, if you go through this little setup right here, we've got a haploid organism. That is the gametophyte. Okay? Down here is the sporophyte that you see at the bottom. Start at the bottom. Let's say we've got a plant. Most of the plants that we have outside that you look at, most of them will be sporophytes. There will be some, such as moss, that will be a gametophyte. The question here is, what is their adult stage? Is it a sporophyte or gametophyte? Most plants are sporophyte. We start right here. Notice with these plants, they form this little extension or extension right there. And then it produces spores. These spores will go through meiosis. Instead of being 2N now, they will be haploid. They will multiply through mitosis. In other words, they will just constantly keep making a large number of them. They will then, through meiosis, after they go through that, we will have eggs, male and female gametes form that'll be fertilized and make the zygote, and it just keeps going through the process. You'll learn a lot more about that whenever you get into 1082. But I want you to remember, alternation of generations occurs in plants. One generation, the sporophyte is diploid, the gametes are haploid. And you will get into this quite a bit when you get into 1082, so I'm not going to cover it a lot in through here. This is another version. The bad thing about alternation of generation, there's not just one version of it. It depends on the organism you're talking about. Plants will have one, different plants will have different kinds. Ferns will have one kind that moss doesn't have. So we're not going to cover all of that right now. That's in 1082, one of the first things you'll start into when you get into 1082, talking about plants. So here's what I want you to concentrate on. I want you to know what alternation of generations is, that it produces two different generations, a sporophyte generation that is diploid, a gametophyte generation, which is haploid. Animals don't do that. Meiosis is what we are going to be concentrating on here. Note, meiosis takes place in two cell divisions. Mitosis, there was only one cell division. That resulted in two cells. We're going to go through one cell division here and get two cells, but then it's going to go through another division and wind up with four at the end of this. So meiosis goes through two cell divisions, mitosis only one. Meiosis 2, mitosis 1. Note these two cell divisions result in 4 daughter cells, and each of those cells are haploid. After the chromosomes duplicate, remember that is going to be an interphase, right? Like we looked at before, that part's not going to change. Remember in interphase, we had the G phase, G1, S phase for the chromosomes. DNA was duplicated, and the other parts of the cell were being prepared. So after that occurs, after chromosomes have been duplicated, you've got two divisions. Meiosis 1. The homologs pair up and separate, resulting in two haploid daughter cells with replicated chromosomes. In meiosis 2, the sister chromatids separate. Each chromatid now becomes a chromosome and will go into an individual cell. The result is those 4 cells. So let's look at what happens. An interphase is where we get the chromosomes replicated. So we started with one chromosome, now they're duplicated. In meiosis 1, you've got these chromatids, sister chromatids. The homologous chromosomes separate. In other words, this pair separates. Haploid cells with duplicated chromosomes. So now we've gone, this was a diploid cell up here that carried 46 chromosomes if it were in humans. Okay? Those pairs are going to separate. So this cell now will have 23 pairs, and that cell will have 23 pairs. But remember, they're still sister chromatids. They have been copied. In meiosis II, they will divide again, and these sister chromatids separate, and we wind up with 4 cells. The blue and the red are representing the chromosome from either the mother or the father up there. If this is chromosome number 1, right up here, pair number 1, you got part from your mother, part from your father. They duplicate, so you've got 2 of each. Now they separate into individual cells. The pair breaks apart. If they don't stay in a pair anymore, one chromosome from your mother goes one cell, the chromosome from your father goes into another. They've been duplicated. So in meiosis II, these chromosomes just separate to make 4 individual cells. Everybody good with that? Yeah, Monique.
So basically, the sister chromatids and interphase, those are basically just the blue ones are separated from the red ones into 2 different cells. That's all that's happening here into meiosis I.
What happens is, if you look, each one of these cells, this is one pair. Let's say it's chromosome pair number 1, one from the mother, one from the father. In interphase, they copy the chromosomes. Now they become sister chromatids. This cell then divides, and you've got the mother chromosome in one, the father's in other. Now keep in mind, this is when we're talking about individuals. This is not fertilization here. This is to produce gametes. Everybody, every one of us, our first pair of chromosomes, we got one from the mother and one from her father. So whenever these duplicate, half of your chromosomes are going to be the ones come from your father and the other half from the mother. Then they go into individual cells. The chromatids separate and become chromosomes. The difference is, this is a diploid cell. It's got a full set of chromosomes. These are haploid cells. Now,
Thank you.
I don't make you go through and understand what happens. I mean, there's 2 different cycles here. What happens in anaphase and all of this stuff. There's one thing that I want you to remember about this, so you don't have to remember every single thing. Note the sister chromatids are genetically identical. Division in meiosis I occurs in 4 phases. I want you to know that. And that is just the prophase, the metaphase, the anaphase, telophase. There's no pro-metaphase. It's just prophase, metaphase, anaphase, and telophase, and cytokinesis. This shows you what happens. There's one thing that I want you to make sure that you understand. Notice up here in prophase. Now these are the same chromosomes that we had seen in that other picture, but now something happens called crossover. Crossover, notice how these chromosomes at certain points will attach to each other. they will actually take this segment and put it over here and that segment over there. And when you do that, you wind up with something that looks like that. Now, notice how these go through metaphase, but they're lined up. Now, they pull apart. We got different sections, if you notice on here. There's a red end onto that chromosome. That's occurring over here. The prominence of that and the importance of it is the chromosomes that you pass on to your children will not look exactly like your chromosomes. This is an example. This is the way that nature does genetic recombination. And that's what it's doing. It's changing the combination of genes that you've got. And because of that, the chromosomes that you give eggs or sperm, your children, are not going to be identical to yours. They will have gone through these changes. If you notice up in through here, what we've started with, this is meiosis 1. Notice here, we're getting ready to go in through cytokinesis, and the sister chromatids are still together. Well, we've got to have a cell that's only going to have a haploid number to it, the egg or the sperm. So that is why we go through a second division. So remember, crossover is occurring in meiosis 1. And it will recombine the gene sequence. This tells you what is happening in through there. Don't spend a lot of time on that. Remember this. And this is in prophase 1. In crossing over, non-sister chromatids exchange. Now notice, it's the non-sister chromatids. These two blue ones are sister chromatids. The two red ones are sister chromatids. But if you take one of this and one of the red, those are called non-sister because they come from two different chromosomes. So it is those non-sister chromatids that do the exchange. Each pair of chromosomes forms a tetrad, or a group of 4 chromatids. Each tetrad usually has one or more chiasmata, X-shaped regions where that crossing over occurs. Up here, if you notice. That's where they attach and where the crossover occurs. I don't want you to remember about metaphase on this. It's not going to be that much different than what happened in mitosis. Same with anaphase, same thing. They pull, the spindle fibers start to pull them towards the post. No different than mitosis. Telophase and cytokinesis, same deal. The thing I want you to remember about these is in meiosis 1, we have sister chromatids. They are not separated. They have gone through change or crossover that occurs in prophase. Meiosis 1 produces 2 daughter cells that have sister chromatids. In meiosis 2, we have these 2 chromosomes that came out of meiosis 1 right here. So they've gone through crossover. So now we've got these 2 cells. They will go through prophase and metaphase just as we've done before. But now what is going to happen, when they get to metaphase, instead of those chromatids being pulled to the folds, once again, go back. Notice here, it is the sister chromatids being pulled to the folds. They're not separated. Now, it will be the individual, the chromatids will separate. It will no longer be a 2N number cell diploid, it will become a haploid cell. So, these sets pull to each side. Remember, these are sister chromatids. Each half set will go towards one end of the pole. At the end, we wind up with 4 haploid cells. There is not that much difference between mitosis and meiosis, but remember this part. At the end of meiosis 1, there will be 2 cells produced that are diploid. In meiosis 1, those diploid cells that have copied the chromosome, they will separate and we will wind up with 4 haploid cells. So basically the same thing happens in meiosis 2 as 1. The only difference is in meiosis 1, when it happens, it's got the copied chromosome, sister chromosome. And in meiosis 2, it doesn't recopy the chromosomes, it just pulls the 2 chromatids apart to make 4 cells. And that is 4 haploid cells. So, the product of meiosis will be 4 gametes. They will be haploid. Everybody understand that? That's the main part of this I want you to understand, right there. You don't have to go through all the phases. They're no different than mitosis. So make sure you understand those. And like I say, you don't have to go through these phases. Here's what I want you to remember. Because we have gone through crossover, this bottom line, each daughter cell is genetically distinct from the others and from the parent cell. Crossover exchanges parts, so they're not identical. So you have got 4 daughter cells produced. Which one will be fertilized is totally random. There's nothing that guides it. This is why you may have some similarities to your brothers and sisters or your parents, but you don't look exactly alike them unless you are a monozygotic twin, which means what happens sometimes when you get to that first cell, the zygote, when it is formed, sometimes it can divide and make 2 zygotes, and then they go through mitosis to make 2 embryos and 2 fetuses. Those are look-alike twins. The zygote is what divides and makes 2. There was a story in the news today y'all will look at. Things are getting ready to change. Chinese are doing gene editing on people now. There is a system that we discovered from bacteria called the CRISPR-Cas system, and it enables people to go in, take an embryo, cut out genes, and put in what we want. So, that's getting into some shaky ground when you get into doing that. Number one, what they do is going to be passed down to other generations from that individual. They do something wrong, and honestly, I don't have the faith in the Chinese science and technology that they're not going to make a mistake. It's a big deal. And this is one of the first times that people have come out publicly and say they're doing that. So, and they do it, you do it in a petri dish. You take the embryo out of the mother, put it in a dish, add certain enzymes and certain gene segments, and you can take part of it out, put what you want in it. and that is, if it don't scare you, it should. Because for this time, they say there are so many people, which was kind of a shock to me. But they said there are so many people that are HIV positive in China, which is not one of the places I would have thought that would be a high problem. But what they were doing is taking one gene out of a person. There is one protein in your DNA that determines can the HIV virus get into your cell or not. You have to have a certain kind of protein. Unfortunately, most people have the kind that allows it. So they're going in to cut out that gene and putting in a version that says you can't. Maybe they'll put in a version that says you have to be 6 foot tall, blonde haired or something. Or maybe they want to make soldiers. Find a few genes that will make them very aggressive. You can do that.
What if the government decide to take advantage of that to manipulate people to their will?
So it's something to be concerned with. That is mitosis. This part compares mitosis. Look at it. Meiosis produces number of chromosomes, reduces from 2 to 1, producing cells genetically different. Mitosis simply clones the cells that are there. This is a chart that shows you that. Once again, I don't want to spend a lot of time on meiosis because it's not that much different. It just goes through a second division primarily, and crossover is the big deal with that. There are a few other things you get into, genetics and cell biology you may cover, but they're not that important for us. All this part, don't worry about too much. On mutations, I want you to know what a mutation is. It's some kind of change to DNA to the code. It can be because something is left out, a nucleotide is left out. It can be because some are added. It can be because they shift from one place to another. But that is also a method of genetic variation. So how can DNA and genes vary? One is by mutations, and the other one is crossover. This reshuffling of the alleles is what they're talking about. 3 mechanisms. I do want you to remember this one. Genetic variation among offspring. Why do offspring have different genetics? Number 1, independent assortment of the chromosomes. When you got those crossed over, you don't know which one you're going to get. So independent assortment. Second one is crossing over. And the third is just totally random fertilization. You don't know what chromosomes, what genes are coming in. So do remember those 3 things. Responsible for genetic variation, independent assortment, crossing over, and then random fertilization. This will tell you about those 3 things. And we've talked about most of it before. This is talking about combinations. Notice here, the number of combinations possible when chromosomes assort independently into gametes is 2n, where n is the haploid number. For humans, n equals 23. There are more than 8 million possible combinations of chromosomes. So we can get a lot of variation. This shows you these different combinations that you can come up with. And when it says combination, it means the one that you get from your mother and one you get from your father. When you're going from haploid to making the zygote. Possibility 1, if you've got this combination, you can come up with these. They're going to be pretty uniform. If you come over here and you've got a different combination, then they're all going to be different. So, much is it. Remember, crossing over results in recombinant chromosomes, which means they are being parts taken away and others combined. This shows you the effect of crossover. Nothing new. Random fertilization means we do this in animals. I mean, we pick out the parents so we can pick out traits to get the traits that we want. In people, you don't do that. It's just random fertilization. So that's that. You should know what to remember from that. Remember, mitosis has one cell division and produces diploid cells. Meiosis has two cell division, produces 4 haploid cells. Now this is the one getting into heredity. This is the dreaded one where you've got to do Punnett squares, and folks, you've got it easy. We also will learn how proteins are made. And this is the big deal. Proteins are called, when we make the copy of the gene, that is called transcription. When we turn that copy into a protein, that is called translation, to read. We will first start off talking about these hereditary traits. Most of you in high school have undoubtedly done some of these things with the Punnett squares. You'll get to do some good Punnett squares here. And you're going to have to do one of those on the exam. It'll be worth quite a few points. So make sure you understand when we cover this, how to do these things. But to start this out, keep in mind something about Mendel. Doesn't tell you a lot about him. I'm going to give you some information. Mendel was an Austrian monk. And this was in, I think, around the mid-1800s. And in these monasteries, these people would go, there'd be a bunch of the monks, and they all would have to do a job around that monastery. Some were carpenters, some took care of animals, some made beer, some made cheese, some was responsible for upkeep, maintenance, cleaning. Others would be copying books. Everybody had a job. Mendel's was to be the gardener. When he was doing this, remember, there was no concept of gene or heredity. No one knew. And while he was studying the peas that they were growing, the peas, the benefit from them is they grow very quickly. You can grow many, many generations in 1 year. It's not like if you were working with an animal and you waited 3 years to go generation to generation. So what he started to notice was that if plants had particular characteristics such as the color of the pea, the color of the flower, the height of the plant, all of these seem to be transmitted to the offspring in some way. He didn't know what that way was or how. So basically, that was the first concept of what a gene is. He didn't call it a gene, but he knew there was something. So he started to keep records and keep track of this when he would grow the plant. And then he started to selectively take particular plants and put them together to see what the outcome was. And then it got to the point where he came up with the idea, well, if this trait's present, it's going to show up. That's a dominant trait. Sometimes the other traits will show up, sometimes they don't. Those were recessive. So that is the way genetics got started. And remember, that was in the 1850s. It was in the early 1900s before the idea of a gene came into being, given the official name. And then it was in the 30s where Watson and Crick got together and came up with the idea these genes are on this molecule DNA. So, it has not been that long. Keep something else in mind. A great deal of what has been done in genetics has been done within easily the last 15 to 20 years. And the reason it being, when people were trying to do all of these things by hand in microscopes, you were limited. Now, with the advent of the biotechniques such as PCR, gel electrophoresis, and most importantly, with computer software programs that can do billions of functions in the time that it would take us to do one with pencil and a piece of paper. It's opened up a lot of doors. Because of that, we wound up making huge leaps in genetics. We actually now, we can take genes, chromosomes, DNA out of a person, multiply it into the billions, cut them in certain places. We can even now map out what every gene is on a human being. Every gene on a human being has been located. We don't know what they all do, but we know where they're at. They are given a name. That came about not that long ago. I think maybe around 2010 or around that time. That is called the Human Genome Project. And the Human Genome Project is, you can go to the site and it's got every chromosome and all the genes listed. Some of the ones we know what they do, it will tell you what they do. The others are blank, most of them. We don't know what they do. It's complicated. We will get into it coming up.